In this chapter, we are going to talk about complex lipids with two really big focuses. One are components that make up a cell membrane, and the other are steroids. So let's talk about the components that make up a cell membrane first. And to do that, we need to talk about phosphoglycerides, which are also known as phospholipids. So those are two terms that are used interchangeably. So these are, like we mentioned, complex lipids, and they're one of the major components of cell membranes. In these compounds, when we look at their structure, and there's an example structure here, what you're going to see is there's a glycerol backbone, and this is just 3 CH or CH2 molecules that are shown here in a line in the pink. And then if you remember, glycerol had 3 alcohol groups. You're not going to see the alcohol groups. These have been attached or reacted with other groups. So these are going to contain a glycerol molecule, and then we're going to see an ester linkage to a phosphoric acid. So we see an ester linkage. Right? And then that's generally linked to some sort of other alcohol, which a lot of times contains an amine. And so what we're talking about with this particular group here is this business right here. So that is this. So we're going to have some sort of phosphate group. Then we are going to see that the two other alcohol groups here and here of that glycerol backbone are generally linked to fatty acids. So there's also going to be a fatty acid component here. Okay. Now, I want to point out a few things. This part here is ionic. So you're going to see a positive and a negative charge here. And so what that means is it tends to be polar and interact with water. And then these parts here are mostly carbon and hydrogens, and so those then are going to be nonpolar. So when we talk about phospholipids, what we're going to see is a lot of times these drawn as a cartoon, which is shown in green. And this a lot of times is called the polar head. And that corresponds to this ionic organic phosphate group right here. And so a lot of times it's called the polar head. And then this part here is called the nonpolar tails. Okay, so when we see phospholipids in a cartoon, particularly in cartoons that show us cell membranes, you can see here are the nonpolar tails, which correspond here, and then here is the polar head, which also corresponds here. Another common phosphoglyceride is lecithin. Before we jump into lecithin, I want to show you a couple components. So the most abundant phosphoglycerides have as one of their phosphate tails either a choline, which is shown here, or an ethanolamine, which is shown here, or a serine, which is shown here. And what you're going to notice is there's ionic groups, and here we've got 2 ionic groups. And so these are the things that most often make up that polar head, right, that are attached to the phosphate group. Okay, and so when that group is choline, right here, so we see how it's attached there. There's where we've got phosphatidylcholine, and we can see this part is the choline part. Okay. every single phosphoglyceride that contains choline is classified as a lecithin. So when we talk about a lecithin, which I have a hard time saying that word, so just bear with me, right? Those are just phosphoglycerides that contain choline. Now, because you can get different fatty acids here at the first and the second position of the glycerol, you can have a lot of different lecithins that are possible. Each lecithin contains a negatively charged phosphate group here and a positively charged quaternary nitrogen. And so, as we mentioned before, the charges make the polar head hydrophilic. So it's the charges there that are really going to make it hydrophilic. And as we've mentioned on the previous slide, these are really going to be very hydrophobic. So this kind of difference in the structure where we have this that's hydrophobic and this that's hydrophilic allows molecules like this to function as emulsifying and micelle forming agents. So these tend to be emulsifying and micelle forming agents. Now, micelles play a really important role in the transports of lipids in the blood, and then lecithin is commercially extracted from soybeans for use as an emulsifying agent in food. So this is used quite a bit in food, and it's going to give the food a smooth texture. So a lot of times you'll see it on the labels of things like margarines or chocolate candies. It's pretty common, and that's just to help it have a smooth texture. Now, another common phosphoglyceride are cephalins, which we see here. Now, when we talked about those individual structures that are found on the phosphate groups on the previous slide, this is going to be either the ethanolamine or the serine rather than choline. So if this group is the ethanolamine, which is not shown, or serine, which is shown here, then these fall under the class of cephalins. Okay, they're found in cell membranes and are particularly abundant in brain tissue. So we find these in cell membranes and brain tissue. They're also found in blood platelets, and they play a really important role in the blood clotting process. Now, again, note the presence of the negatively charged phosphate and carboxyl groups and the positively charged nitrogen here. And that characteristic also, like the lecithins, helps it function as an emulsifier. So this can also be used as an emulsifier. Now, another lipid that's part of the cell membrane are sphingolipids. When we look at sphingolipids, right, these do not contain a glycerol. So no glycerol, right? And instead, they contain sphingosine, which is this molecule here. And that's just a long chain unsaturated amino alcohol. So we talk about the long chain. This is the long chain here. And we've got an amine group here, and that's where the amino part comes from, and then the alcohol here. And what we find is that the amine will attach to a fatty acid, and then the alcohol will sometimes attach to, per se, one of the phosphate groups. And so this is called sphingomyelin, which is shown here, where we have the long chain here, and then the amine is attached to a long chain through an amide bond here. And then we also see the difference in charge here. So this is shown for you as a cartoon here, but then also as the structure here. Now, in sphingomyelin, the choline that's attached to the sphingosine is through a phosphate group, and there's a single fatty acid, of course, that's attached to the sphingosine. Now, so I want to put this into writing. So that sphingomyelin, that contains a sphingosine, a fatty acid, a phosphate group, and a choline. Okay. Now, large amounts of these sphingomyelins, like shown here, are found in brain and nerve tissue, and in the protective myelin sheath that surrounds nerves. Now, a second type of sphingolipid is a glycolipid, and this contains sphingosine and a fatty acid, so similar to the sphingomyelin. But instead of the phosphate group with the choline, it contains a carbohydrate unit. And so let's look at those. So this is an example of a glycolipid. When we look at this, we see there's the same long chain. Fatty acid is here, and it's got that amide linkage. And then now we have a carbohydrate here. And then, of course, here's the cartoon for this. Glycolipids are often called cerebrosides because of their abundance in brain tissue. Right? And these do not contain a phosphate. Now, when we talk about sphingolipids, there are several human diseases that result from abnormal accumulations of sphingomyelins and these glycolipids in the body. And they are shown here on this table. Each of these diseases is a result of an inherited absence of an enzyme needed to break down complex lipids. So each disease is the result of an inherited absence of an enzyme needed to break down these complex lipids. Now, when I say these complex lipids, I mean sphingolipids, right? So that's what we're talking about when we use this word. Now, we're going to kind of shift gears and we're going to talk more about how lipids fit into a cellular picture. And so to do that, we need to review cell structure just a little bit. So when we talk about cell structure, I just want to remind you there are two different cell types that are found in living organisms. There's prokaryotic and eukaryotic cells. Prokaryotic cells are things like bacteria and cyanobacteria. They're smaller and they're much less complex than the eukaryotic cells. So prokaryotic cells are things like bacteria. Okay. Now, both types of cells are essentially tiny membrane enclosed units of fluid that contain the chemicals involved in life processes. Eukaryotic cells also contain small bodies called organelles, which are suspended in cellular fluid, which we know as cytoplasm, right? And so we also know that organelles are the sites where we have a lot of specialized functions in eukaryotes. And so I've got a list of the most prominent organelles and their functions on the next slide, but that's one of the big differences here. Now, both of these have a membrane, and that's really what we're focused on. And the membrane performs two really vital functions in organic organisms. So the external cell membrane acts as a barrier between a living cell and its environment, and then an internal membrane surrounds some of the organelles and creates cellular compartments that have separate organization and functions. So, two roles of the cell membrane. The first one is you can have an external cell membrane that functions as a selective barrier between the living cell and its environment. And then the second function is you can have internal membranes that surround organelles and that creates a cellular compartment. That has, I said have, but I really want to say has there, that has separate organizations and functions. Now, when we look at a cell membrane here, what we're going to see is these are our phospholipids. And so this is called a phospholipid bilayer, where bi here just means two. So you have two layers of phospholipids. And the way they organize themselves is the nonpolar regions are what interacts. So this is our big hydrophobic tails. So we've got our hydrophobic tails that interact here in the center. And then our polar heads here are oriented with the aqueous environment here outside of the cell, and then the aqueous environment inside of the cell here. So the membrane then can act as a barrier. Now, I promised you a table with some of the cellular organelles. These are things that you've probably learned all through kind of your science career, particularly in biology. So just as we go through and reference these, like if I say a nucleus, which we're going to talk about particularly when we talk about DNA and RNA. This is a really great reference where you can say, oh, yes, I remember that the nucleus is in the cell, and that contains the hereditary material, and that directs protein synthesis. So these are really great to review as we're going to start talking a little bit more about chemicals in cells. Now, let's talk about membrane structure again. Most membranes contain about 60% lipids and about 40% proteins. So when we look at a cell, so here is our cell, here is the nucleus, here's the cytoplasm with all the organelles in it. And we are looking at a membrane right here, kind of shown in green outside. And if we were to come up here and zoom into this membrane, what we're going to see is everything in green, those are your phospholipids. So this is our glycerophospholipid bilayer. We've pointed out the hydrophobic region, and then we've got those two hydrophilic regions. Then what you're going to notice, right, is that we have some things that look like this, and these are proteins. You can have a protein that's just on the surface, or you can have a protein that bridges through either side, and things are going to kind of pass back and forth. We also have some carbohydrate side chains here. These are like the ABO blood markers that we talked about. Then what you're going to notice here is you have cholesterol that's embedded as well into the cell membrane, and we'll talk about cholesterol in just a second. So the model that describes this cell membrane particularly is called the fluid mosaic model. And this is just kind of what we described. The lipids are organized into a lipid bilayer, and there are hydrophilic groups on the outside and hydrophobic groups on the inside of that bilayer. So the fluid mosaic model is just the model of membrane structure where proteins are embedded in a flexible lipid bilayer. And what we find is the hydrophobic parts of the chains extend toward the inside of the bilayer, and the hydrophilic chains are oriented toward the outside. And so when we say towards the outside, we know that that's means either, right, the outside of the cell or inside of the cell. So when it says towards the outside, we're talking about towards the outside of the lipid bilayer. When that lipid bilayer is broken and the tails are exposed to water, they get really repulsed by the water, and that causes the bilayer to reform. And so that whatever break it is will seal almost spontaneously. Now, membrane lipids here will usually contain unsaturated fatty acids that fit into the bilayers more loosely. So membrane lipids usually contain unsaturated fatty acids that fit more loosely in the bilayers than saturated fatty acids. This is because they generally have a U shape, and so they spread out a little bit. And so what happens then is that increases those unsaturated fatty acids, and I abbreviate fatty acids as FA, right? Increase the flexibility or fluidity, it's described both ways, of the membrane. And so because of that, what happens is some of the proteins in the membrane kind of float in that lipid bilayer like icebergs in the sea, okay? Whereas others then are going to extend all the way through. So these are like our little icebergs that are floating along, and these extend all the way through. And so the lipid molecules then move kind of back and forth through all of this. And so that's why it's called a fluid mosaic. It's fluid because everything is kind of moving here. And then it's a mosaic because you've got a lot of different pieces. And so that's where that fluid mosaic model comes from. So this ends kind of the first part of our screencast where we said, hey, we're going to talk about membrane structure and the lipids that make up membranes. And then in the second part of our screencast, we are going to talk about steroids. So steroids are just a broad class of compounds that contain 4 fused rings in this particular pattern. So there's 1, 2, 3 cyclohexanes and a cyclopentane here. And every steroid has these rings. Now they may have different things attached, but that's what they're going to have. These are soluble in nonpolar solvents, which is why they're classified as lipids, right? And really important ones that are included here we're going to talk about are cholesterol and bile salts, and we're also going to talk about sex hormones. So let's talk about cholesterol. So cholesterol is the most abundant steroid in the human body. Because we already saw that it makes up part of cell membranes. So it's a really essential component of cell membranes. And is a precursor to other really important steroids, like vitamin D, bile salts, male and female sex hormones, those types of things. Now, we find that cholesterol is synthesized in the liver. And it's found in the foods we eat. Cholesterol has received a lot of attention because of the correlation between its levels in the blood and the hardening of arteries. So this is still an active research area, and it's not really known the role that's played by cholesterol in hardening the arteries. But it's now considered really advisable to reduce the amount of cholesterol in foods we eat to kind of help with some heart health. And then also reducing the amount of saturated fatty acids in the diet also lowers cholesterol production in the body. And so those of us who have high cholesterol, that is what we are advised to do. Now, we talked about cholesterol being a precursor to different components. One of those is bile. So bile is a yellowish brown or green liver secretion. So bile is a yellowish brown to green liver secretion. and it's stored and concentrated in the gallbladder. Okay, and so what happens is when you have partially digested fatty food in the small intestine, that causes the gallbladder to contract and empty the bile into the intestine. And so the bile itself doesn't catalyze any digestive reactions, but it is important in lipid digestion. So fats just overall are more difficult to hydrolyze than, say, starch or proteins because they aren't soluble in water. And so in the watery medium of the digestive system, fats tend to form these really large globules with limited surface area exposed to be attacked by those digestive juices. So the chief constituent of bile are bile salts and two waste components, the cholesterol and bile pigments. And those help kind of spread out the fats. So they act a lot like soaps. They're going to emulsify the lipids and break the large globules into many, many smaller droplets. And then after that, right, the total amount of lipids is still the same, but now we've got a much larger surface area for hydrolysis reactions to digest them. So I want to put some words to that. So when partially digested fatty food enters the intestine, the gallbladder contracts and empties bile into the intestine. Now, bile acts like soap and emulsifies the lipids. And so it breaks them into smaller droplets. Right. And then there's a larger surface area available for the hydrolysis reactions of digestion. Now, a second function of the bile salts is also to emulsify cholesterol found in bile. So excess cholesterol is concentrated in the bile and is passed into the small intestine for excretion. If cholesterol levels become too high in the bile, or if the concentration of bile salts is too low, the cholesterol will precipitate and form gallstones. So gallstones are just the precipitate of cholesterol. Okay, and so gallstones are generally 80% cholesterol, and right are colored by entrapped bile pigments. I don't want to, so I've got a picture of the bile salts, the gallstones here, But I also wanted to point out, I forgot to earlier, that this is a principal component of bile salts. So this is the principal bile salt. So that's kind of the major one there. Now, the passage of a gallstone here from the gallbladder down the common bile duct to the intestine causes really excruciating pain. And sometimes one or more of the stones will become lodged in the duct, and that will prevent bile from passing into the small intestines, and then fats can't be digested normally. If a person's affected with this, they feel a lot of pain and they become really nauseous and ill. And so then what you have to do is, if it becomes really serious, you have to have your gallbladder removed. And that will also remove the stones. So, that is one of the steroids that we're going to talk about. There's another steroids that we need to talk about, hormones. A hormone is just a chemical messenger secreted by specific glands and carried by the blood to a target tissue. Okay, and then it triggers some sort of response. Right? There are two categories of hormones that we're going to talk about. We're going to talk about adrenal corticoid hormones as well as sex hormones. And so here are some examples of the adrenal corticoid hormones. Now, when we talk about the adrenal gland, these are just small amounts of tissue located at the top of each kidney. The outer layer of the gland is the adrenal cortex, and that produces a number of really potent steroid hormones, which are our adrenal corticoids. So these are produced by the adrenal cortex. And these are classified based on their function into two groups. The first group are mineralocorticoids. These regulate the concentration of ions in bodily fluids, right? So, for example, aldosterone, which is here, increases absorption of sodium and chloride and is involved in the water balance in the body. Okay. Then another example, well, the second classification are glucocorticoids, and these just enhance carbohydrate metabolism. And an example of that is cortisol, which is shown here. And what that does is it increases glucose and glycogen concentrations. And has anti-inflammatory effects in the body. Right? Cortisol and its ketone derivative, which is cortisone, which we saw in the previous slide, are really, really powerful anti-inflammatory agents that are used in the body. And so if you are given like a steroid like prednisone, these are used to treat inflammatory diseases like rheumatoid arthritis or asthma. I've ended up having eye drops that are like this to help treat some swelling in my eyes due to allergies. So these are pretty commonly prescribed to help with that. Now, the last thing we're going to talk about in this screencast are sex hormones. We've got two kind of classes of sex hormones that we're going to talk about. We're going to talk about sex hormones that are found in men. These are produced in the testes, and in women, these are produced in the ovaries. These help secondary sex characteristics appear at puberty are the reason they help develop these. And so when we talk about the male sex hormones, big ones include testosterone, and the structure showed here. Again, we can see that steroid ring, and then we also see this as the secondary male sex hormone. Then when we talk about female sex hormones, these are things like estrogen and progesterone that cause different changes to the uterus. And again, I want to point out, you can see the same structure that you would see in any of the other steroids here. And with that, that ends our discussion in lipids. As always, if you have any questions, please feel free to pop by my office.